Greetings, everyone. I am Michael Adeshokon from IIT Nigeria. I have been nominated to present the main highlights and project findings on Gari&Eba product profile for work packages 1 to 5. Yes, it's a collaborative work with contributions from partners working on EBA as the product profile both in Nigeria and in Cameroon. This shows the roadmap of interaction between the work packages from identification of quality perception by consumers in WP1, which is communicated to WP2 to translate into lab measurable traits. The traits are delivered to WP3 to develop a high throughput protocol for assessment. The traits are delivered to WP4 to include in the new breeding programs and afterwards tested by users for validation. The outcome of survey activity 3 shows that women are overrepresented among the poor and underrepresented among the richer farmers. Women have less access to land and are rarely landowners. Women dominate in processing role. No significant gender difference with regards to preferences, and more attention for processing and food quality in breeding will benefit women in Nigeria and Cameroon. Summarizing preference survey results conducted in Nigeria and Cameroon in order of importance. These preferred characteristics are the outcome of focus group discussions and individual interview conducted in Nigeria and Cameroon. The preferred characteristics for cassava roots includes large size, high dry matter, mature roots, not rotting, and white color. Also for Gari, which is an intermediate product, the user's preference include white or butter color, shiny, dense or heavy, and swelling when making Eba. And of course, for Eba, the major consumption form of Gari in Cameroon and Nigeria is mainly characterized by its textural properties, smoothness, non-sticky, must be stretchable, moldable, and no traces of darkening or browning. Also for participatory processing activity 4, processors prefer fresh cassava roots that is heavy, large root size, gives good food quality, high dry matter with white cream or yellow color. The Gari, shining color with homogeneous granule size and good taste, loose on the fingers, then crunchy. The desired characteristics for Eba are smooth, it should be moldable, stretchable, white, cream, or yellow color, easy to swallow, and moderately soft or hard. On processing the root to Gari, processors desire high Gari yield with no discoloration, easy release of water during pressing with little fibers in the mash. The consumer quality criteria for EBA activity 5 in order of importance are generally the desired quality, the color and the textural properties as well as good odor. Projections for period 4. WP1 will continue to engage WP2 in providing feedback on the most important traits that are identified, both for processing to Gari, the appearance, texture of Gari, then appearance, texture of Eba, as well as odor. For work package 2, activities planned for period 3 are involved to standardize the protocols for EBA preparation at laboratory scale level, to develop SOPs for textural profile analysis of EBA using the texture analyzer, to develop SOP for sensory textural profile analysis and conduct sensory and instrumental texture profile analysis using the developed SOP. Also, biophysical analysis carried out on the focus traits provided from WP1, as well as analysis on Gari produced using 18 UYT cassava genotypes and characterized for focus traits. The focus trait as advised from World Package 1 for Gari are color, swelling power, bulk density, dry matter, then starch. Also, for EBA, basically these are textural properties involving moldability, stickiness, hardness, and cohesiveness. To standardize the protocol for preparation of EBA, this was done in close collaboration with the NRCRI team. 100 grams of Gari was weighed into a bowl and reconstituted in 300 ml of boiled water. In Nigeria, the preference for EBA differs across region. For instance, in the southeast region, soft EBA is undesirable compared to the southwest. Therefore, for the southeast, 250 ml of boiling water was used to reconstitute the same quantity of Gari. The constituted Gari is allowed to stay for about 60 seconds to gelatinize, and then a planetary dough mixer was used to mix the EBA to have a consistent dough. The EBA was immediately transferred into an aluminum foil and properly labeled to control the temperature, and then placed in a warmer. Six cassava genotypes of contrasting cooking qualities were used to prepare the EBA. These genotypes were selected based on biochemical analysis conducted on 18 genotypes of UYT cassava provided from the breeder, and result of the survey from WP1. These informations were clustered, and from the clustering analysis, we were able to select two good, two best, and two poor cassava genotypes, which were used to prepare the EBA for the development of the SOP. We developed SOPs for sensory and instrumental texture profile analysis of EBA. The EBA prepared from the six selected cassava genotypes of contrasting cooking qualities was used for the SOP. The stable microsystems texture analyzer was used for the analysis, and coupled with standard compression plating with 30 mm in diameter. The test speed of 2 mm and trigger force of 5 grams was used for 2-cycle compression test. The shape and geometry of sample is important in instrumental textual analysis. Therefore, a plastic open-ended cup of 2.2 centimeter height and 3.6 centimeter in diameter was used for sample collection and presentation. Therefore, each sample had a uniform dimension for each test. 6 replicate measurements was taken for each sample, and the SOP was submitted for peer reviewing. Also, 12 trained panelists were allowed to evaluate the same 6 EBA at the same time with the texture profile analysis, and the sensory attributes were hardness, smoothness, moldability, and adhesiveness. A scale of 0 to 10 points was used for the sensory evaluation, and the temperature of sample for both the texture analysis and the sensory analysis was 35 degrees C. Both SOPs were submitted for peer reviewed and validated. The perspective for period 4 for WP2 is to conduct biophysical analysis of the focus traits provided by WP1 on the new set of cassava genotypes that will be provided by WP4 and the Gari produced from it. Also, instrumental and sensory textural profile analysis will be conducted on the new set of materials. In period 4, we intend to increase the number of genotypes that will be used both for the instrumental and the sensory profile analysis. to see if we are able to establish correlation between the sensory profile analysis and the instrumental textural profile analysis. Also, we are going to conduct proof of concept on the effect of polyphenol oxidase and non-enzymatic branding on quality of Gari&Eba in collaboration with CERAD. We are also going to develop proof of concept on the relationship of amylose content and pectin on some textural attributes of EBA, such as stretchability. For WP3, the planned activity is develop SOPs for dry matter and color measurement on blended cassava. Three SOPs were developed and validated in period 3. Also to transfer SOPs for intact root, blended root, and color into the new RTBfoods template. Collection of spectral data of Gari produced from 18 cassava genotypes provided by the breeders. A total of 144 spectral data comprising of 18 clones in 2 replications and 2 locations were collected in duplicate. Also, a calibration model was developed for dry matter content on intact root using the NIS, and the R square of 4.96 was obtained. Also, a calibration model was developed for the focus biochemical functional traits on Gari. Prediction model was developed for swelling power, dispersibility, starch, and sugar. As earlier mentioned, 3 SOPs were developed in period 3. I will make reference to 2 of the SOPs that are related. The SOP for color and dry matter measurement in fresh blended cassava roots. The fresh cassava root is divided longitudinally into 4, and the 2 opposite sides were pulled and chopped into small pieces. The small pieces were quartered, and 2 opposite ends were further pulled to have a representative of each root. An electric blender was used to blend the fresh yam and transferred into a transparent clean petri dish. This was then transferred to the benchtop NIS for dry matter measurement, at the same time using the chromometer to measure the color. Repeatability study was also conducted on the SOP by calculating the root mean square error for 10 replicates reading of the blended samples, and the RMSA range from 5745 to 26899 with an average of 12745. The standard deviation was less than 1, and the minimum of duplicate readings was collected. Also in period 3, several calibration models were developed, and one of them is calibration feasibility for functional properties of Gari. In 2019, about 129 Gari samples were prepared from the mother trial provided by the breeders, and calibration models were developed for some functional traits like swelling power, solubility, sugar starch, and dispersibility. The calibration model were improved in 2020 by adding other trials into the populations, and the model were improved for most of the traits. For instance, solubility was the R square for prediction was 0.70 in 2019, and in 2020 it improved to 0.84. The same for sugar, which was 0.76, and in 2020 it improved to 0.88. Prediction performances for the traits are improved when the number of sample increases to 201 in 2020. Also in period 3, for NRCRI, 117 spectra of unmilled Gari from 13 clones at UYT stage at Otobi location were collected using the ASD, and calibration model was developed for dry matter and starch yield on the intact roots, where R square is about 0.52 and 0.54 respectively. Also, calibration model was equally developed for starch and amylose content on the unmilled Gari, with R square of 0.66 and 0.67 respectively. This will be improved upon in period 4. The perspective for period 4 for WP3 is to develop a calibration model using the NIS for starch content on intact fresh cassava roots. We have done for dry matter, so we want to develop also for starch content on intact roots. Also, we will explore the calibration feasibility of functional analysis of milled and unmilled Gari on the focus traits provided by WP1. Of course, we would expand the needs database of cassava genotypes using the new materials that will be provided by WP4 in period 4. Also, would expand the needs database for milled and milled Gari using the new set of materials. Calibration development attempt will also be done on textural properties of Eba. And we have installed the hyperspectral imaging camera in the lab in IITA, and in period 4 we would attempt to apply the HSI for measurements of quality traits in cassava roots and products. For work package 4, both IITA and NRCRI collaboratively evaluated 67 clones across 4 locations, generating Gari productivity data. A total of 72 genomic selection cycles were evaluated in replicate trials across the 4 locations. For the broad sense heritability, the set A trial at Ikene and Ubiaja had the highest heritability estimates for Gari yield, while the trials at Agowu and Ibadan had a moderate broad sense heritability, which is an indication of genetic control. Also, for the trait correlation, Gari yield, which is the Gari weight, is positively correlated with dry matter, and it has a weak negative correlation with fresh root yield. Heritability estimate for Gari yield also at NRCRI shows that high heritability values exist across trial, indicating a strong genetic control, and the range of heritability for Gari yield is between 0.64 to 0.7. For the impact of G by E interaction, we observe a highly significant genotypic responses across environments for Gari with strong crossover interactions among the traits. And GGE results also indicated G by E interaction for Gari yield, where the 2PC accounted for 74% of the total phenotypic variations in the Gari. In WP3, there is a very close interaction between WP4 and WP2 in terms of exchange of ideas and the processing of EBA. Also, all the materials being used, the UYT 36 clones, the NCRP trial from Ibadan and Ikene were provided by WP4. Also, the breeding program at IIT and NRCRI have contributed to WP1 and WP5 through the provision of advanced breeding cassava clones for testing under the mother, baby, and tricot trials across different states in Nigeria. The achievements of WP5 in period 3, tricot trials in 4 states with a total of 320 farmer households have been harvested and results have been analyzed. Results show that post-harvest trait and especially food quality trait determine the final choice of a variety. Also, 2 WP5 trials with advanced clones and varietal popular among farmers have been established to be evaluated with communities in period 4, one in Osun State and another in Benue State. Multiplication of advanced clones has been done for transportation of clones to Cameroon for WP5 work in Cameroon, littoral zone for Gari and Eba, and the eastern zone for for fufu. The main insight on Gari&Eba from participatory processing using pairwise ranking methodology. The preferred characteristics based on 276 pairwise ranking of roots, Gari and Eba products coinciding with activities 3, 4, and 5 results. For fresh roots, high dry matter, high product yield, high root yield, large size, easy to peel, bright white, softly when rated. For Gari, bright color, uniform granule size, swelling when making eba, crunchiness, heaviness, good taste, oil acceptance, and for eba, good appearance, not dark in color, heavy, smooth, stretchable, moldable, firm, and good taste. The image shows the Gari prepared from some of the genotypes and the color before and after roasting. The same goes for eba. Identify varieties for calibration of good and bad constellations of lab measurement on food product. Preferred newly developed clones for Gari&Eba based on pairwise ranking of root and Gari&Eba. So these are the genotypes that were preferred, included in local variety with clustered genetically with one of the improved varieties of IITA. Then the non-preferred improved varieties because of the texture and color. These are the traits. The important to keep to calibrate bad color for branding is the main negative trait. Projection for period 4. Second year tricot trial will be established. 21 replanted year, one tricot trial will be fully monitored by food scientists to develop food science anchoring of second year tricot trial. Processing evaluation with champion processors in Osun and Benue State using the advanced clone. Samples of best and worst clones to the lab and inform WP2 on calibration of textural data. Status of identified priority quality traits in breeding. For processing to Gari, the traits of concern are foaming and discoloration, which are not accessed at the moment. For Gari, we have product yield, bulk density, woody filament, and shyness of the color, which are at the moment well accessed. And for the product, we have volume expansion, modability, discoloration, and odor. These are in progress at the moment, and the proof of concept for period 4 includes polyphenols in the roots, dry matter in the roots, and G by E effect, lignification, whose effect is not yet identified, then low polyphenols in the roots. For the products, proof of concept on amylase pectin interaction, as well as proof of concept on the presence of polyphenol. Thank you very much.